pictures of all of you, just some of you. And if you're curious why I've drawn you, check out EcoGenome on Twitter. Or come talk to me later. I just love to communicate. This has been such a great meeting, I have to say. It's been so fantastic having a chance to meet many people here, and I just love the positive, warm attitude of folks at this meeting. So yeah, it's just a big honor to be invited here, so thank you so much, Jackie. So I am here to talk to you about my favorite plant-biotic interaction, which is the interaction between plants and the microbes that inhabit floral nectar. So again, thank you for that shout out earlier. This was a really great setup for my talk. So I'm going to tell you about kind of one key interaction between natural history and what our lab's really focused on, ecological theory. And we are really interested in using field experiments and also just observations in the field to inform kind of broader questions about how we think about community processes. And the system that we use are these nectar-inhabiting microbes. So today I'm going to frame my talk in this trifecta of understanding natural history, ecological theory, and how we use experimentation both in the lab and in the field to address some of these really fundamental questions. So we're going to go from natural history to experimentation to ecological theory. So first, let me tell you a little bit about how these guys work. Our main focal plant is Diplocus aurantiacus, or the sticky or bush monkey flower. These are shrubby monkey flowers. I know we have a lot of monkey flower enthusiasts here in the audience. yeah. These guys are shrubby though, so they're not the little ones. They're like these giant bushes. And they're pretty well distributed throughout the coast of Northern California, and we mostly have field sites around us in the Bay Area. And we mostly focus, though, on the microbes that inhabit their nectar, mostly bacteria and yeast. So if you like kombucha, any kombucha lovers in the audience here? Yeah. So here's a shout out to a local kombucha company, Pacific Culture Kombucha. But if you like kombucha, you know about SCOBY, which is the symbiotic culture of bacteria and yeast. And you know that depending on what kind of bacteria and what kind of yeast you have in your SCOBY, it really changes the taste of that kombucha. And so you can think about nectar as tiny SCOBYs that have both bacteria and yeast living in them, and the kind of ratio of bacteria and yeast and what kind of bacteria and yeast you have, as well as the kind of community, the type of nectar that the plant is making, really influences how that nectar tastes to pollinators. So this morning we heard a great session about the interactions between plants and pollinators. And so what I hope to convince you today of is that nectar microbes are a really third interaction in this tripartite and not bipartite interaction, where not only are they impacted by plants and pollinators, but they actually impact plants and pollinators. So how does this work? Well, there's a bunch of microbes around in different flowers' nectar, but also, inside of plants, in the roots, and in the air. And they get deposited into floral nectar, mostly by pollinators. So Diplocus aurantiacus is mostly bird pollinated, which we also heard about this morning, by Anna's hummingbird, and sometimes also by bees and other pollinators, but mostly hummingbirds. And so on the beaks of birds, or the tongues of birds, or the mouthparts of bees, they deposit microbes into newly opened flowers. And so each flower constitutes a discrete microbial community. Now, a lot of our research in our lab has focused on these species interactions in nectar. Oops, I'm going to use a lightsaber here. So you might imagine that we have a flower where yeast gets inoculated alone. And we have another flower where both yeast and bacteria get inoculated. So as those flowers grow, those microbial communities also grow, you might get different end communities right before that flower senesces. So the flower that started out with just yeast might have a lot of yeast after a couple days, and the flower that had both yeast and bacteria might have fewer yeast. And so what this suggests is that what we've observed is that bacteria suppresses the growth of yeast, right? This would be a conclusion from this. And so when we go out in the field, we can harvest flowers, we can take out nectar using tiny glass straws, microcapillary tubes, and grow them on agar plates, or we can do next-gen sequencing and see what the microbial communities are. So I'm going to orient you to some field data. So I'm going to show you a plot where on the y-axis is bacterial density, and on the x-axis fungal density. And each point is going to represent the microbial community of an individual plant. So if we see points over here, it's low and high densities of bacteria. Points over here is low and high densities of yeast. And any points in the middle are flowers that have both yeast and bacteria. And what we see is we either get plants that are dominated by bacteria or dominated by yeast, but very rarely, if ever, get flowers that have both bacteria and yeast. And so what this suggests is that bacteria and yeast might be negatively affecting one another inside of flowers. So, okay, that's all fine and dandy, but as a botanist, why do we care about this? Well, the microbial communities have really big impacts on plant fitness. We've seen that bacterially dominated flowers have reduced seed set, they're less attractive to pollinators, and there's less nectar that's consumed by pollinators. So you might think of a bacteria-dominated flower as kind of a not-so-tasty kombucha like a salty dog. Sorry if you like salty dog. And this is contrasted by yeast-dominated flowers, which pollinators seem to like more, and there's some research that suggests that yeast might actually make a volatile cue that's attractive to pollinators for those flowers. So the outcome of this competition between bacteria and yeast really can have important implications for this tripartite interaction between plants, pollinators, and the nectar microbes. So, okay, this is cool. Single flowers, microbe competition, changes pollination. But it's not just single flowers, right? Because flowers are on plants, and plants are embedded in a landscape. And so when we think about these communities, it's really important to consider that these are a landscape of flowers that are connected by dispersal via pollination. And so our lab's done a lot of work on this question, for example, bagging or caging flowers or plants to exclude pollinators, and going around the entire Bay Area, all the way from Bodega Bay down to Big Sur, and looking at how microbial communities change across this very large gradient. And so we can address a lot of different questions using the system, such as how connectivity influences species abundances and distributions, how nectar microbes might influence pollination across landscapes, and whether nectar microbes might even influence plant evolution. So you might be wondering, but how? That leads me to the second part of this talk, where I'm going to talk about the parallel lab and field experiments we can do that I think is the really important and novel aspect of the system. So as I told you before, we can do lab experiments where we go out into the fields, we can pick flowers, we can grow microbes on agar plates in the lab, we can do sequencing with them, we can exclude pollinators, and kind of get a sense of what's going on in the field. But once we have interesting hypotheses, it's really easy to do experiments in the lab as well, where we can grow nectar microbes on agar plates, we can grow them in fake nectar, we can make in the lab, and put them in little tubes that represent flowers, right, floral communities, and we can move them around like pollinators would using pipettes. And so we can make very replicable experiments between the field and the lab. So this was the data I showed you before from the field, and this is a similar experiment we did in the lab, and we see very similar patterns of this competition between bacteria and yeast. Right, so remember that there's this negative interaction between bacteria and yeast. And so I want to tell you a little story about how this can inform how we think about ecological theory, specifically rapid evolution. And it starts with an observation we've been making for several years in the fields. And that is, in the early spring, when our flowers start blooming, we see that most of the communities are dominated by bacteria. Most of the flowers are bacterially dominated. But by the end of the summer, when the flowering season is ending, most of the plants are actually dominated by yeast. And so I wondered, could the yeast be evolving to be less affected by bacteria? Or to frame it in kind of a more theoretical way, could rapid evolution of microbes change the strength of competition between microbes that, as I just told you, has really, really important implications for the fitness of the plant and the behavior of pollinators? So to address this question, this really tantalizing question in the field that it's quite hard to address in the field, it's hard to see rapid evolution, we decided to turn to laboratory experiments, where we harvested flowers from the field, we grew microbes on agar plates, and we isolated individual species so we could do experiments with them. Now, I'm going to be telling you a very yeast-focused story and how bacteria will negatively affect yeast. And what we did with those isolated yeast from the field is we used experimental evolution, where we put single clones, so genetically identical individuals, into nectar that simulated early arrival by a bacteria. So we can make this nectar in the lab, and what I did was I grew bacteria in that nectar, and then I filtered the bacteria out. So it looked like there were bacteria there to the yeast, but there actually weren't. And I passaged them from tube to tube to tube over 2 months, which is the length of the flowering season of our plants, simulating those microbes, seeing bacteria multiple times as they move flower to flower. And so at the end of 2 months, we had these evolved yeast. So remember, we started out with isolated yeast from the wild, and in the end, we had these yeast that were evolved. But instead of calling them isolated yeast, I'm going to call them ancestral yeast in this talk. That doesn't mean they came from millions of years ago. It means that they're ancestral relative to our experiment. And so once we had this ancestral and evolved yeast, we did competition experiments competing them against the same bacteria to address the question, which yeast competes better against bacteria? The ancestral yeast that we got from the field at the beginning of the season, or this evolved yeast that should be better at competing against bacteria? And so we did these experiments where we grew them together, and then we measured the density of both bacteria and yeast to ask how well did the yeast grow with bacteria, comparing ancestral and evolved. So we're measuring, I'm going to show you points that represent the final yeast density with bacteria. And what we see is that the evolved yeast grew much better in the presence of bacteria than the ancestral yeast, suggesting that we could do evolution in the lab that provides insight into these phenomena we see in the field. So as we think about changing species abundances and distributions, even over the course of the entire season, or just a season, the changes that we see might not be because of static competitive differences, but because of evolution that happens in extremely short time frames. And that could be what's happening when we see bacteria-dominated flowers at the beginning of the season and yeast-dominated flowers at the end of the season. And so the rapid evolution of nectar microbes might have really important implications for plants and pollinators as we move from nasty tasting nectar at the beginning of the season to much nicer tasting or more attractive nectar at the end of the season, which again might have really important implications for the seed set of plants and their reproductive success, as well as even plant evolution, and for pollinators in terms of their behavior with those volatiles. So just to recap what we learned here, we used a field survey where we observed that there's more yeast in flowers as the season progresses. We did an experimental evolution project to test this in the lab to see if this holds up. And what we found is that rapid evolution can change competitive dynamics of microbes over the course of a single season. So what I hope is that if you're really into systematics, or if you're into plant-pollinator relationships, or you just care about plants, you take away from this talk that nectar microbes play a really important role in mediating plants' interactions with the world in terms of their reproduction, as well as a takeaway that no matter what species interaction you care about, those interaction strengths can change over very rapid timescales because of evolution. And with that, I'd like to acknowledge the wide variety of people who helped with this project, a huge team of undergraduates who did a lot of this work, as well as undergraduates in a lab course who did collected a lot of the preliminary data for this experiment, folks in my lab and other collaborators, as well as the organizers of this fantastic conference. So thank you very much.